Today, 20th January 2022, MNT second year, subject is biochemistry. So, topic is yesterday we are discussing in glycolysis pathway in metabolism. So, energy balance in glycolysis 10 ATP are produced, 2 ATPs are consumed, net yield of ATPs is 8. This is the last pathway in energy balance in glycolysis. So, the next topic is the TCA cycle, tricarboxylic acid cycle. The original name of this cycle, citric acid cycle, is a citric acid cycle. It was postulated by Krebs in 1937. He was postulated by Krebs in 1937. TCA cycle is a cyclic sequence of reactions. TCA cycle is a cyclic sequence of reactions. Cyclic means ఒక చర్య ఒక రౌండ్ గుండ్రంగా ఉండేటటువంటి చర్య మాదిరిగా ఉంటుంది అన్నమాట. Reactions in a pyruvate formed glycolysis completely oxidized carbon dioxide and water. Pyruvic is a last component of glycolysis, a pyruvic acid. So, steps in tricarboxylic acid cycle. Acetyl coenzyme A oxaloacetate stakes to water by the enzyme of citrate synthetase is going to citrate plus coenzyme A. This is the first reaction. Once again, acetyl coenzyme A plus oxaloacetate plus H2O by the enzyme of citrate synthetase. It gives rise to citrate plus coenzyme A. And then converts to citrate by the enzyme of aconitase. It gives to cis-aconitate by the enzyme of aconitase. It gives to isocitrate. Then isocitrate plus NADP plus by the enzyme of dehydrogenase, alpha ketoglutarate plus CO2 plus NADPH plus H plus. So, fourth reaction, alpha ketoglutarate plus NADs plus CoA, coenzyme A. CoA means is a coenzyme A. Succinyl coenzyme A plus CO2 plus NADH H plus. Fifth reaction is succinyl coenzyme A additional phosphate plus GDP by the enzyme of succinyl coenzyme A synthetase. It gives to succinate plus GTP plus coenzyme A SH. Then next reaction, succinate plus EFAD by the enzyme of succinate dehydrogenase. It gives to fumarate and EFADH2. Fumarate plus water by the enzyme of fumarase L-malate. L-malate plus NAD, L-malate dehydrogenase. It gives to oxaloacetate plus NADH plus H plus. So, this is the oxidation of valmate to oxaloacetate takes place in this step. It is catalyzed by L-malate dehydrogenase. Oxaloacetate can combine with another molecule, acetyl coenzyme A starts another cycle. Clear? So, once again, I will read the steps one by one. Acetyl coenzyme A plus oxaloacetate plus water by the enzyme of citrate synthetase. It gives rise to citrate plus coenzyme A. This is the first reaction. Then second reaction, citrate by the enzyme of aconitase is gives rise to cis aconitate. By the enzyme of aconitase, it gives to isocitrate. This is the second reaction. And other one, third reaction, isocitrate plus NADP by the enzyme of dehydrogenase, alpha ketoglutarate plus CO2 plus NADH plus H plus. Clear? This is the third reaction is completed. Fourth reaction starts alpha ketoglutarate plus NAD plus coenzyme A. It gives to succinyl coenzyme A plus CO2 plus NADH plus H plus. Clear? Fifth reaction, succinyl coenzyme A plus phosphate plus GDP. Succinyl coenzyme A synthetase. Succinyl coenzyme A synthetase. It gives to succinate plus GTP plus coenzyme A SH. Sixth reaction, succinate plus EFAD by the enzyme of succinate dehydrogenase. Fumarate plus EFADH2. Then fumarate plus water by the enzyme of fumarase L-malate. L-malate. L-malate plus NAD, L-malate dehydrogenase oxaloacetate plus NADH plus H plus. NADH plus H plus. So, this type of reactions are especially used for this tricarboxylic acid cycle. Then steps in urea cycle also. Urea cycle, very important cycles. One is formation of arginosuccinate, and second one is L-citrulline aspartate, formation of arginine, formation of urea. So, especially in three steps, one is formation of arginosuccinate, L-citrulline plus aspartate plus ATP by the arginosuccinate synthetase enzyme, arginosuccinate plus AMP plus PPI. Second, formation of arginine succinate by the enzyme of arginosuccinate lyase. It gives to arginine plus fumarate. Then third one is formation of urea, arginine plus water. It gives to ornithine plus urea. So this type of three reactions is completed of urea cycle. Conclusion, especially first pathway is glycolysis, second is TCA, tricarboxylic acid cycle, third one is urea cycle. Urea cycle, formation of arginosuccinate, second formation of arginine, formation of urea. Clear? These three steps only. Once again I will tell you, formation of arginosuccinate, L-citrulline plus aspartate plus ATP by the enzyme of arginosuccinate synthetase, arginosuccinate plus AMP. Clear? Second reaction, formation of arginine. How to formation of arginine? Arginosuccinate by the enzyme of arginosuccinate lyase, it gives to arginine plus fumarate. Third one is formation of urea, arginine plus water, it gives to ornithine plus urea. So these are three steps by the completed of the urea cycle also. So very important cycles in TCA or glycolysis and urea cycle also. This is three cycles are very important in examination purpose. Any one cycle is given by the annual examination. So you have to ready the three glycolysis pathway and tricarboxylic cycle and urea cycle. Urea cycle is especially only 3 steps. Don't forget it. Okay, so especially in short questions, what is glycolysis, what is the name of glycolysis, what is the defined glycolysis, glycogenesis, and define urea cycle, what is TCA, what is urea cycle. Long questions, write about signification of TCA cycle and explain significance of glycolysis, write the pathway. So this is the very important in long questions. So thank you all of you.